CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats, or simply a bunch of short DNA segments that are spaced out. Before we go into its effects, we need to figure out what that actually means. CRISPR was not something that someone randomly thought of someday in a lab. It was actually first discovered in bacteria. On the genome of the bacteria, there are Cas genes which are transcribed and translated into CRISPR RNA and tracer RNA and the Cas enzyme. The two types of RNAs combine into a single guide RNA. When bacteriophages inject their DNA into the bacteria, the Cas enzyme will break the bacteriophage's DNA up if it has the short palindromic repeat in its genome so that it can use to bind to the bacteriophage's DNA. If it doesn't, the Cas enzyme will add it to part of its genome with the rest of the spacers so that it can use it in the future.
In order to edit the genome, CRISPR uses 3 different methods. One of these methods is disruption. The Cas enzyme reads the DNA strands for the protospacer adjacent motif, or PAM for short, which is just a specific strand of DNA that Cas9 is able to read and initiate the cleaving of. The Cas9 enzyme has a gRNA, or a guide RNA, that will bind to its complementary DNA strand once it has been identified by Cas. Once it is bound, the Cas enzyme will cleave the DNA through natural repair processes. A few nucleotides will be lost or added from the gene, which will most likely result in its inactivation. Another of these methods is deletion, in which the Cas9 enzyme contains 2 gRNAs for 2 cuts to cut the whole DNA segment out. For example, sickle cell disease is caused by a 1-point mutation in the gene that encodes for hemoglobin. Using deletion, the single faulty base pair can be removed and replaced so that the amino acid is correct. Deletion can also be used for longer strands of DNA. The Cas enzyme can also add it in a DNA segment after the cut, which is called insertion. The way this works is that the Cas enzyme takes in the RNA, or in a lab setting, the plasmid DNA, which is then inserted into the cell and into the cell's genome by cleaving the plasmid on the correct spot. So that it is just a linear strand. It then uses the enzyme transposase to align with the Cas9 enzyme to cut the DNA and then insert the plasmid DNA.
Through these genome modifications, CRISPR can have a lot of positive impact on the medical field. For example, back from Unit 4, we learned about how p53 activates cell cycle arrest in cells during the GO checkpoint. However, when it is deactivated, oncogenes are free to undergo mitosis, which is cancer. Using CRISPR, you can insert a correct p53 back into the genome to stop oncogenes from undergoing mitosis and reactivating the ability to perform apoptosis. However, because of this great power to change the world, there are some major implications of CRISPR's use as a bioweapon.
CRISPR can be applied to develop viruses for humans. Much like some fungi in ant populations, many believe that viruses or some other microbe can be used to control humans through the use of CRISPR technology, pretty much zombification. These zombie fungi for ants can be genetically engineered for humans. These fungi release chemicals that reach the nervous system of the ant, so the ant moves up the desired conditions of the fungi and then creates a stalk so that its spores can continue to spread to the other ants. This idea is a little bit far-reaching, so bringing it back to something more realistic is the development of viruses and bacteria that are feasible for humans. Just like with plants, bioterrorists can bypass most of the human genome's resistance from the existent viruses like COVID, polio, or malaria. By changing the genetic code of these viruses, bioterrorists can edit the DNA so that the body would not be able to perform any immune response because the body would be unable to detect a new variant. On top of this, these viruses can be made more viral and deadly compared to their first introduction to human population. All of these scenarios are very hypothetical, but how close could some bioterrorists be to coming up with a CRISPR-made virus, and how can we prevent it? Could CRISPR actually be used as a bioweapon? Because CRISPR hasn't been around for a long time, the costs associated with changing multiple genes to create a bioweapon is still huge. Kasevegi, A recent sickle cell anemia treatment released to the public would cost around $2.2 million for treatment. This high cost for the development of this CRISPR system to change one gene is simple compared to the CRISPR that would need to change several genes at a time to have bioweapon capabilities. The cost for the population of a gene like that right now would be well in the billions. From both of these factors, the public has no major threat to CRISPR-edited microorganisms or CRISPR because it's still in early development. Thank you for listening and have fun with our lab.